Welcome to Characterization of Extracellular Vesicles with Nanoscale Flow Cytometry, presented by BioCompare and sponsored by Beckman Coulter Life Sciences. My name is Tamlin Oliver, Managing Editor of BioCompare, and I'll be the moderator for today's webinar. George Brittain is a staff advanced research scientist at Beckman. He'll be sharing his knowledge with us through a presentation as well as a Q&A session that we hope you'll participate in. We are thrilled to be hosting this forum, and we'll get started shortly, but first we want to go over a few housekeeping items. You can submit a question for the Q&A session at any time using the Q&A box on your screen. Just type your question in the blank field and hit submit. In addition, your console is fully customizable, and we encourage you to move, maximize, or minimize any of the tools on your screen to optimize your viewing experience. If you experience technical difficulties at any time, there's a help button on the bottom toolbar of your screen. Ping us, and we'll get right on it. George works in a variety of areas, ranging from inventing new technologies to conducting basic science research. One of his current focus areas within Beckman Coulter involves optimizing and innovating both instruments and sample preparation methods to improve the detection, resolution, and immunophenotyping of extracellular vesicles, viruses, and other biological nanoparticles. Welcome, George, and please take it away.
Thank you for the introduction. I'm George, and today I'm going to talk about characterizing extracellular vesicles using nanoscale flow cytometry. In this presentation, I will demonstrate how to set up a CytoFLEX for violet side scatter detection, considerations for proper small particle experimentation, how to characterize instrument sensitivity, and finally, how to prepare and analyze extracellular vesicles in blood and plasma. Extracellular vesicles are small, naturally occurring cell fragments that range in size between 30 and 1,000 nanometers. They're present in all bodily fluids, and their potential for use as disease biomarkers is the subject of active research in major therapeutic areas. While flow cytometry is the method of choice for characterizing most complex suspension samples, EVs are very difficult to detect by conventional flow cytometry. However, the CytoFLEX is not a conventional flow cytometer. The CytoFLEX is a novel semiconductor-based flow cytometer that has unparalleled sensitivity for both fluorescence and light scatter detection. It combines LED lasers with Wavelength-Division Multiplexing and Avalanche Photodiodes to both maximize signal and minimize noise. The results that are displayed in this presentation will be mostly from a CytoFLEX as flow cytometer, but the small particle principles are universal. Wavelength-Division Multiplexing is a novel system that's designed to parse light wavelengths with maximal efficiency. This highly sensitive system was adapted from fiber optic technology used in the telecommunications industry. By eliminating traditional filter trees, less light is lost due to the inefficiencies associated with dichroic mirrors, and this results in a greater than 95% signal recovery. This is combined with Avalanche Photodiodes. Avalanche Photodiodes are silicon-based semiconductors that use electron hole pairs created by incident photons to produce an avalanche of electrons within the diode. This results in a highly sensitive detection of photons, which are directly proportional to the quantity of incident photons. APDs multiply signals similar to photomultiplier tubes, but with higher quantum efficiency, allowing for better detection of dim signals. Finally, the optic system of the CytoFLEX is modified to enhance signal capture and captures roughly 110 degrees of orthogonally scattered fluorescence and light, scattered light. This increases the capture by roughly twofold over traditional optics. And furthermore, the method of focusing the light reduces background by reducing cross-mixing of signals, which result from the alternate lasers. Some examples of the detection using light scatter on the CytoFLEX are shown in these two figures. So the figure on the left is forward scatter versus side scatter, and the figure on the right is violet side scatter versus side scatter. Forward scatter on the CytoFLEX is not a traditional small angle scatter detection mode. It is actually a comparative signal analysis method, which is referred to as axial light loss detection. And in fact, this is very similar to transit photometry that's used in astrophysics. This system is optimized for the 500 to 50 micron range, 500 nanometers to 50 microns, and it is mostly independent of the refractive index, which can be seen in the figure on the top left. So it is, you can see 500 nanometer particles versus 1,000 and 2,000, which is mostly proportional to the actual size of the particles, as opposed to the refractive index, which you can see in the side scatter detection. Side scatter on the CytoFLEX is much more sensitive, but it's attenuated actually for use with cells and other large particles. It's optimized for the range of roughly 200 nanometers to 20 microns. As an alternative, violet side scatter is available to take full advantage of the sensitivity of the CytoFLEX. And violet side scatter enables the resolution of 80 nanometer polystyrene and 100 nanometer silica beads. Later in this presentation, I'll demonstrate the sensitivity for biological particles. Fluorescence sensitivity on the CytoFLEX is also quite extraordinary. So in literature, the CytoFLEX has been said to have higher Q values than any PMT-based flow cytometer that has been tested. Internal testing and external testing have demonstrated METS-F values as low as 1750 molecules, and the perfect linearity in this range suggests that it should go even further. 100 nanometer 50 labeled polystyrene particles are demonstrated in these two figures at the top, and they can be seen to be easily resolved in both scatter and fluorescence. And a variety of flow cytometers cannot even detect these particles. Next, I'll discuss how to set up violet side scatter detection. The first step for setting up violet side scatter detection on the CytoFLEX involves setting up the detector configuration. The detector configuration can be found in the Cytometer tab, wherein you'll find a series of fluorophore options and filter options, and you can actually assign them to the different channels. The default option on the CytoFLEX is every channel is assigned fluorescence. However, you have to pick one or another and then ultimately assign it as the violet side scatter and assign it with a 405 filter. Once you're done with this, you save the configuration and set it as current. So the next step is to physically swap the filters in the WDM pods. The way that wavelength-division multiplexing works requires filters to be in each slot for the light from a preceding filter to reach a filter in the subsequent slot. Therefore, if you're missing a filter, even in the first slot without an APD in some WDM pods, the signal will not make it to the subsequent channels. And a similar issue, if you put a duplicate filter preceding your filter of interest, you may lose the light in that filter, or at least most of the light. So for instance, if you put a 450 filter in the first slot, and then in a subsequent slot you have 450 looking for PAC blue signal, all of your PAC blue signal is lost in that first filter. So it's important to make sure that you have your filters set up properly. Okay, so next, make sure to change the event rate setting to high. The default setting for the event rate in the CytoFLEX is actually a broader pulse window that's used to set up the laser delays and also the laser alignment if they're really out of whack. However, the broader pulse window actually increases the background noise. So changing it to the high setting actually reduces the pulse window and therefore reduces background and also further improves the signal processing. And it enables more events to be detected and processed. Next, set the threshold to violet side scatter. So even though the detector configuration is set up for violet side scatter, unless you trigger on violet side scatter, the violet side scatter detection does not actually take place. So it's important to set violet side scatter as a trigger and then adjust the threshold setting as appropriate for your sample. The instrument always triggers on height. If you set area, it actually back calculates height, so it's more precise to make sure and actually adjust the height. The specific setting will depend in part on the sample, depend on the gain setting, but it also may vary just a little bit with different instruments, so this will need to be optimized by the user. In general, the optimal threshold setting is roughly tenfold whatever the gain setting is. So if your gain setting is 400, the optimal threshold is generally around 4,000. In the figures above, I have gain setting of 400, but the threshold at 3,000. This is one of my preferred settings, but you can actually see that if you were to adjust on the violet side scatter height scale to 4,000, it would actually be a more optimal setting with less noise. Some optical noise is useful as a baseline. However, it's good to keep it minimal. Below your threshold is actually an optical noise range. Finally, it's important, if not critical, to make sure that your sample probe is cleaned. Debris from samples and buffers actually will build up in the probe, and then this can later break away during acquisition. And due to the nature of small particle detection and characterization, any debris is just sheer noise in your system, so it's important to clean this up. This can be relatively easily done using a panel of bleach, flow clean, a cleanse solution, and then follow that by water to actually flush out the detergent. Generally, this is sufficient within one round or so, but you may need to repeat a second time if necessary. And if major noise remains after that, you most likely have dirty buffer or sheath. It's good for small particle work to deep cleanse your system regularly, and the only fix for dirty sheets is to switch your sheets out to clean sheets. So it's important to take note of this. Less sensitive flow cytometers, if they cannot detect 100 nanometer particles or less, this may not be an obvious situation if you've not experienced it before. Okay, so additional notes on violet side scatter detection. If you've previously identified the optimal settings for your cytometer, then you can bypass step E. The CytoFLEX actually is very consistent between experimentation, and any previously identified settings will actually be optimal in the future, all things being equal. If you haven't identified your optimal threshold, then it makes it very difficult to actually assess the cleanliness because the scales automatically adjust to the sample. You can correct this essentially by clicking on the fit to axis and unchecking the box within the plot settings. However, it's still important to know where to look. Another note is regular CytoFLEX sheet actually works very well for small particle detection on the CytoFLEX, and higher purity sheet is not necessary. And indeed, higher purity sheet is quite expensive, and it's not going to give you much advantage. And furthermore, the CytoFLEX sheet contains an antimicrobial agent that will prevent instrument contamination. So if you are using DI water or something, that if contamination starts to accumulate in your filters or in your sample lines, then you may end up counteracting, the efficiency of any cleanliness or any cleansing process that you do. Considerations for small particle experimentation. So one of the first and probably the most obvious is to prepare the samples with clean buffers. Any debris in your sample buffer will actually be detected as small particles, and it can interfere with your analysis of your small particle samples. So buffers, of course, can be filtered. However, you'll find that the debris are generally smaller than the filter. In fact, in the plots above, 0.2 micron filtration would only eliminate debris roughly greater than 10 to the sixth. And these samples are a variety of distilled water from grocery stores, which seem to be sometimes used by the small particle experimenters. And then finally on the right is DI water. So these compared to HPLC water, you can see that HPLC water just has significantly less debris and contamination. And as it says below, HPLC water is typically the cleanest, and also fresh RO water from a clean system is good. However, RO systems can become contaminated over time. So it's good to use a clean system or just buy the HPLC water. It's important to optimize your sample concentration. If you are analyzing particles, if the sample concentration is too high, you'll start analyzing swarming and other coincident artifacts. When the concentration is significantly high, you'll have an increase in both the height measurements and area. And if it's marginally high, generally the height measurement may be pretty similar, but area can be still significantly high. And if you see in these plots on the far left, one example is the doublet populations that you can see in between the different histogram peaks. As the concentration is decreased, such as over towards 1 to 40,000 in this case, and 1 to 80,000, these doublet populations go away. And then you have the population peaks decreasing in numbers, but not changing any intensities. So these are the optimal settings. One thing that can help is to analyze the abort rate. So the CytoFLEX enables you to see the number of aborts that are occurring in your system, and it should be under 10% for small particles, but under 5% is best. And indeed, you could see in the two plots on the right, you have an abort rate of 3.47% and 2.76. So these are the optimal settings here. As mentioned, the abort rate will definitely help find the optimal dilution, but it's particularly helpful with complex samples. So if you're analyzing something such as EVs, the samples tend to be multimodal if you're analyzing in plasma and blood. They have broad distributions. The population means may be intermixed with noise. And in fact, they may be intermixed with lipoproteins and other debris. So it's good to keep it under 10. Under 5 is best. When under 10, as you can see on the top right, the population means are very, very consistent between dilutions. But however, once you get above 10%, such as the top two histograms here, the 12.47% and 13.82%, you can see the height measurements begin to increase. So this is not appropriate, and at the point that you have swarming, you're not analyzing real events, but rather multiple events passing through at the same time. So General concentrations that are optimal for blood tend to be around 1 to 1,000 to 1 to 4,000. And Ison fractions, if you analyze Ison purified samples, each pass through the column seems to reduce the concentration roughly tenfold. So the numbers indicated in the top right actually reflect this. Another consideration regarding concentration is that it's a balance of the concentration and the sample rate. So the sample is passing through a flow cytometer, the flow cell, and every cytometer has an optimal event processing rate, essentially. And this depends on, the ability of the instrument to actually analyze, but also on the rate that the particles pass through the flow cell. And this is inversely proportional to the particle size. So smaller particles, more can pass through in a given time period than large particles. The sample rate, slow, medium, high, et cetera, the 10 microliters per minute, 60 microliters per minute, is essentially an instrument-based dilution directly into the flow cell. So it has a proportional dilution factor, and it counterbalances with whatever the sample concentration is. So in fact, if you have a very high concentration, and it is optimal at, for instance, 10 microliters per minute, if you increase the flow rate to 60 microliters per minute, it in effect increases that concentration sixfold, so you may induce swarming in your sample. So you need to balance the flow rate and sample concentration. In general, slower rates are better because the core stream and the CVs will broaden at higher rates. And just a side note that this is not related to the time of flight. So I've heard some individuals suggest that changing the sample flow rate affects the speed of these particles passing through the flow cell, but it's completely unrelated. A demonstration of the effect of the sample rate on the CVs of these particles can be seen in this plot. So this is the CVs for violet side scatter height and violet side scatter area for a mixture of 80, 100, 150, and 300 nanometer polystyrene particles at different flow rates. And you can see that the CVs increase as the flow rate gets beyond roughly 60 microliters per minute. Around 90 microliters, there's a marginal increase, but generally around 120, it seems to go up about 2 to 3-fold. Not too bad. In fact, it's much better than some cytometers, but nonetheless, the optimal flow rate would be slower, and then generally between 10 microliters to 60 microliters. But again, lower flow rate, higher concentration would be the best due to the narrower core stream. A final consideration for small particle experimentation: some samples tend to be particularly tenacious, and they'll stick within the sample probe or sample lines, and not just show up, in subsequent or future analyses, but actually can show up directly in the next sample. So in this case, in the top set of charts, you can see the detection over time of events. First, just after washing with PBS, and then running human plasma, and then followed by running just a well of PBS, and you can see there's actually some carryover in the initial 20 seconds or so of the sample acquisition. So this can be addressed by better flushing between the samples if you're reading really dirty samples, and or possibly cleaning. And an alternative, if you don't have time to do this, you can actually gate the spurious events out on the time axis. Now I'll discuss the instrument sensitivity. So here is a plot of violet side scatter detection on the CytoFLEX using a mix of polystyrene and silica beads, including some fluorescent beads. So as you can see in the lower plot, it's indicated where a typical microparticle analyzer, their sensitivity limit, and then also a typical flow cytometer sensitivity limit. So most flow cytometers have specifications for detection of 200 nanometer particles, which you can see here. In the case of the CytoFLEX, we're able to get down to roughly 60 or 70 nanometers. There's at least a decade of sensitivity in excess of even traditional microparticle analyzers. Within this scatter range, you can detect a variety of particles. Here, we show the 80 nanometer particles. Also, right on the cusp is 60 nanometer, which can be detected, not fully resolved from baseline separation, but it is detected. We can perfectly detect and resolve 100 nanometer silica particles with either refractive indices of 1.43 or 1.46, and also a variety of viruses. In this case, you can see 90 nanometer adenovirus, which is quite impressive. characterizing the biological sensitivity is a little more complex. Man-made particles can be used to compare the sensitivities of different instruments. However, their refractive indices don't directly correlate to biological particles. Biological particles actually can have a variety of refractive indices depending on the size. So a population within a population of extracellular vesicles, you may have a whole series of refractive indices due to different concentration ratios of macromolecules versus water. Characterizing the biological detection sensitivity requires the samples to first be purified and analyzed by an alternate means, such as dynamic light scatter or nanoparticle track analysis, in order to first determine the mean or refractive index. And then these characteristics can be correlated to the scatter intensity. Some groups are actually working on algorithms to directly convert scatter intensities to size and or the refractive indices based on mean theory and taking into account a number of instrument variables. Here you can see EV detection in whole blood. So EVs can be detected directly in blood and plasma. On the top, you can see forward scatter versus side scatter for these particles, and you can see where these guys fall out. So on the top left are RBCs. The middle is the forward scatter for platelets, and then at the top right, you can barely see a little bit of WBC. So in this case, analyzing all the way down to lipoproteins and vesicles, the WBCs become a completely rare event. So, you may only see 10 or 20, although interestingly, they have this little miniature forward and side scatter plot. In the bottom, you see violet side scatter versus CD950. So these are CD950 labeled extracellular vesicles, and they also label platelets because these are platelet-derived extracellular vesicles. So, and this is directly in blood, so it's quite sensitive. However, to analyze the size, it works best with monomodal size distributions with very narrow distribution. So here is one method that was used, and there's a variety that can be done, but one method that was used to prepare the extracellular vesicles for testing the threshold sensitivity. In the second plot here, you can see this is whole blood. Subsequently, the samples had been centrifuged to isolate the platelet-poor plasma, removing the platelets. In the fourth plot, you can see the platelet-poor plasma having been filtered through a 200 nanometer filter. So this removes all the large particles, including the platelets. And then finally, these samples were passed through an ISON column, and then 200 microliter fractions were collected in order to minimize and eliminate particles less than 70 nanometers, the pore size of these ISON columns. So this actually effectively narrowed the size range to 70 to 200 nanometers. And interestingly, the different fractions after the exclusion volume had slightly different sizes, which enabled a characterization of a size range. So this can be seen in this slide here. So on the left, you can see the overlay of the violet size scatter height histograms for these different fractions, ISON fraction 5, 6, 7, 8. And then also on the right, the same samples were read using a DeltaMax Pro dynamic light scattering system for Beckman Coulter. And you can see the size of these fractions. And 5 is on the right, 8's on the left. So it goes from 170, 140, 120 down to 80 nanometers. So surprisingly, these readings were in direct correlation with each other, and they indicate that the CytoFLEX can detect EVs at least as small as 80 nanometers. So in this chart, you can see the violet side scatter intensity versus size for the different particle fractions. And the top dashed line here indicates the sensitivity limit of a typical flow cytometer. The second dashed line indicates the typical limit of a microparticle analyzer. And the range beneath this is the extra sensitivity range that you can find on the CytoFLEX. Interestingly, all of these ISON fractions fall directly within this size range and have a perfect linear distribution, so 0.98, 0.99. And it would suggest that if this trend were to continue, that the intensity in the range of 60 nanometer particles would be roughly 30 to 50 nanometers for the EVs. The refractive indices for these particles were actually calculated using inverse approximation and mean theory, and were found to be approximately 1.38 for the 177 nanometer particles, and up to 1.53 at the 78 nanometer particles. So this actually comports well with literature. In fact, the figures above are from Gardner paper demonstrating their analysis using NTA and MATLAB of the different refractive indices versus size, and it is almost exactly. So you look at 177, it would be expected to be around 1.38, and 78 would actually be somewhere around 1.5. So it's an interesting correlation. Now, analyzing EVs in blood and plasma. So this at this point gets a little bit deeper because there's more protocol type notes. So preparing EV samples, once you're set up, you're confident in what you're looking at, analyzing EVs can actually be as simple as just staining the samples and then acquiring them on the flow cytometer. Staining whole blood, analyzing it on the flow cytometer. They can be analyzed in blood and plasma, and they can also be further purified. In some cases, it may be better to analyze in plasma. In other cases, it may be better to purify. Depending on the purification method, some populations can be eliminated. Centrifugation, of course, and filtration will eliminate larger populations. Size exclusion columns will eliminate smaller. And so populations eliminated may be totally expected, or it can come as a surprise if you don't expect that your particle is a little larger than expected or a little smaller. So it actually can provide important characteristics. PEG precipitation, on the other hand, can have variable recovery and actually precipitates a lot of protein in addition to samples, so it's just something to consider. All methods generally have some contaminating lipoprotein if you're analyzing it from blood, but any reduction versus the amount that's in whole blood is helpful because lipoprotein is one of the primary noise populations. If combining multiple methods, such as filtration and size exclusion columns, you may eliminate a little bit of your population here, a little bit there, and then ultimately, when you analyze the samples, what you're looking at or intending to look at is not actually there. So thoughtful sample prep is important. And, you might actually need to cater a specific isolation approach to the population of interest. So showing some example plots of the different steps of purification from the above purification that I discussed. I want to talk about a couple of the characteristics. So in the left plot, it's actually whole blood, analyzing, in this case, CD9 EVs. So you can see I'm pointing to the smallest population, not smallest, but the lowest population on the CD9 and violet side scatter plot. So this tail that extends out from noise appears to be, should I say, almost exclusively lipoproteins. I mean, there's EVs, there's some exosomes, but if you check the population percentages here, RBCs, which are typically 1,000-fold higher than WBCs, 100-fold higher than platelets, in this case are only 4% of this plot. The lipoproteins are multifold higher in proportion than RBCs. And in fact, these actually stain with a variety of APO markers, but I can account for maybe 15 or 20%. I'm assuming there's additional APO markers that may label that have not been tested. This population can actually vary between donors pretty dramatically, and especially after someone eats. So when they eat, they got a lot of lipoproteins. In the purification process for platelet-poor plasma, this demonstrates even gentle preparatory centrifugation, such as a 200 times G centrifugation, not only starts separating out the platelets and RBCs, but it actually starts dipping down into the extracellular vesicle population. So if one of your populations of interest here is in the, somewhere between 200 to 500 nanometer range, depending on how you prepare the samples, you may be eliminating this population. And furthermore, at the point of 200 nanometer filtration, this population gets completely eliminated. So it might be something to consider, and if you're looking at that population, use a larger pore size. There's actually some interesting multi-parametric immunophenotyping for populations in that size range. Finally, on the right, you can see the eyes-on column pretty dramatically reduced the lipoprotein fraction here. So this is actually quite useful and increased the relative proportion of CD9 extracellular vesicles in this case about 6-fold over plasma. So this is actually a decent approach. However, as noted, some populations may be better to analyze directly in the blood, or you may lose them. So minimize handling in some cases. And also, if you have time, resources, and sample, it'd be good to perhaps compare multiple techniques side by side. Staining EV samples is pretty simple, more or less. Generally, there's a variety of markers that are used to analyze extracellular vesicles. The most common are tetraspanins, obviously population-specific markers, homing markers, and other activation markers. Some antibodies work perfectly well for staining EVs, and others don't. And it can vary for a variety of reasons, including, different clones may, just one works well, another doesn't. Same clone from different vendors may have different efficiencies. And sometimes even the same clone with different conjugates from the same vendor one can work, one cannot. In general, flow cytometry antibodies are optimized for cells, and a lot of times these antibodies have different concentrations in different samples, in different vials. So you may be looking at a dramatically different staining range. So this could be one factor contributing to that. Finding the best antibodies can obviously take a little trial and error, and the ones that work will need to be titrated. If the antibody has aggregates or a lot of fluorophores, the best thing to do is to filter or centrifuge. But if they're really noisy, such as some antibodies, they don't actually filter the fluorophore after conjugation, you may simply just need to use another antibody because these guys can have a lot of noise. Samples are labeled by adding the antibody cocktail, mix them together, optimal concentrations, and then incubate with the sample in the dark at room temperature for approximately an hour. They can, of course, be stained longer, but there's not much advantage, especially beyond 2 hours. And then less than an hour can actually result in poor staining, so the samples may not be as well, they may not have as good of a stain index. After staining, the samples can be simply diluted and analyzed on the flow cytometer, depending on the initial concentrations and final concentrations. Probably somewhere in the neighborhood of a 1 to 100 dilution would be about the minimal to minimize, collecting antibody noise and stuff, and reducing non-specific background staining. But for whole blood, plasma, the optimal concentration ranges are typically 1 to 1,000, 1 to 4,000. So these dilutions are generally sufficient to reduce antibody noise. We don't need to do a special purification method. PFA included in your buffer actually can help better in the case of EVs than with cells, at least more noticeably. If you have, for instance, a 96 well plate full of samples, by the time the samples get to the 96th well, after diluting 1 to 1,000 or 1 to 4,000, a lot of the antibodies can dissociate, so the intensity may reduce a bit for the final samples. If you include PFA, it will reduce this effect. The particular dilution ratio for your sample was discussed in part 2, but it's good to acquire a dilution range. So if every time you run the samples, if you run it 1 to 500, 1 to 1,000, 1 to 2,000, if it turns out that your 1 to 1,000 sample is much too concentrated, you have backup data to ensure optimal results. And finally, one method for actually purifying these EV samples after staining, because it can be actually quite complicated, is size exclusion columns. So this will remove the free antibodies by getting them trapped in the pores and then eluting your sample. But it's not necessary in most cases, or at least in a lot of cases. And it actually, as mentioned, can affect your population recovery. So thoughtful sample prep. Some example staining of platelet-derived EVs here can be seen. So comparing whole blood versus ion fraction 6 from the samples demonstrated above. So the first set of plots on the far left is just demonstrating RBCs versus what I call not RBCs. The only reason for this gate is that RBCs, in some cases, and a couple of these antibodies have a little bit of background intensity, so you'll see a little nub of a population sticking off. So just to eliminate confusion there, I removed those guys. Platelets are left in because I'm analyzing platelet-derived EVs, and this is a decent control population, essentially, to identify, that they're actually staining platelets. So the primary things to look at here are CD9 versus CD61. CD61 and CD9 are both platelet markers, and as you can see, not only does it stain the platelets, it stains the platelet-derived EVs that stick out from the population. And it's quite specific. If you look at staining with CD63, there's minimal to no staining here. CD63, it does not typically stain a lot of populations. However, it does. I mean, there's some that come in there. It's more of a rare population. And then CD81. So it's very specific here now to CD9 and CD61. And indeed, if you stain with CD41, it'll also triple label this population. In the bottom panels, this is ion fraction 6. So you can see there's actually a much higher preponderance here of the CD9 and CD61 platelet EV population. And in fact, it pulls out what appears to be a little bit of CD81 staining for a roughly 10% fraction of the sample. There are a variety of EV populations that are present in the blood, and they're variably present actually in different donors. So the main populations tend to be quite consistent between donors. The numbers may vary a little bit here and there, a little more, a little less, a little bit higher staining intensity, a little less, but they're not always the same. And some are, in fact, quite rare. Like maybe 1 in 100 donors, although some are, higher ratio, 1 to 10, 1 to 5. One method for controlling for specificity of EVs people use is to lyse the samples with detergents. So detergents will target the membrane-bound vesicles and presumably don't target antibodies and other components. So it can help confirm that, in fact, what you see sticking out from the noise is EVs and not just a tuft of antibody aggregates. Two of the most common are Triton X and SDS. In the case of whole blood, detergent lysis is a little bit more complex. So whole blood has a supernumerary of red blood cells, tons of lipoproteins and all this stuff. So it's not going to lyse exactly the same as a purified cell culture derived EV sample, but it can be done. I mean, it just takes maybe a twofold more detergent or so. However, no matter how you dilute this, it actually will increase the noise of the sample if you're triggering by light scatter. So detergents create their own micelles. They can incorporate with lipoproteins and even EVs and increase the relative size of these fractions. So it is perhaps not the best approach for whole blood, but it can be done. Other controls for specificity are listed here. So, and there's certainly more, but here's just a list of 7 of them. So you can use isotype control antibodies to test for nonspecific background binding. You can use alternative antibody clones to the same marker to demonstrate redundant specificity. So, while one antibody may bind to the EV nonspecifically, two to the same marker specifically binding to the same will show that it's, or at least support that it's a true population. Analyzing buffer and the antibody cocktail alone will actually demonstrate the presence or absence of aggregates. Some antibodies actually do have aggregates, in particular some of these PE antibodies. Serum plus antibody cocktail can help check for aggregation in the serum. It, in my opinion, isn't super likely to happen, but theoretically it's possible that different reductive capacities of the serum or something may alter the antibody binding. If there is actual aggregation in the serum, the aggregates would presumably be fluorescent in all channels. You're not going to aggregate specifically 50 antibodies together and then aggregate specifically PE antibodies. It would be clumping all these guys together, so it would kind of be a broad signal, kind of like apoptotic cells. Superfiltering the plasma to remove all EVs larger than the cutoff can actually help demonstrate the specificity for EVs in the actual sample, rather than aggregation or something else. And for example, using a 20 nanometer filter will eliminate most EVs. It won't eliminate exomeres, but most EVs, because they're typically 40 nanometers and up, 30 nanometers and up. However, one thing to consider, plasma is very difficult to filter to small pore sizes. And in fact, if you put plasma through a 0.02 micron filter, you may only get like 50 or 100 microliters out on the other side. So you might end up running through a few filters if you're trying to collect a larger sample fraction. But since these samples need dilutions of 1 to 2,000, 1 to 4,000, you don't need a large volume of sample to actually test it as a control. Another control would be the actual presence or absence of specific population labeling for different population markers for the population. So an example would be labeling platelet EVs with multiple platelet markers like CD9, 41, and 61, but not with other markers like CD161 and CD 63. Because if you got nonspecific labeling, it's not going to nonspecifically label only with the population markers of interest. I mean, of an actual population. So this actually can very well demonstrate specificity. And then finally, certain membrane dyes will label only membrane or presumably only membrane-bound vesicles rather than proteins and other things. So this can be used to verify that you're actually analyzing a vesicle population. However, dyes and stains tend to add a lot of noise to the samples if you're analyzing by side scatter. And it can actually be improved potentially by filtering, but even filtering sometimes isn't enough. So finally, my summary. In summary, the CytoFLEX flow cytometer is very sensitive and capable of detecting EVs at least as small as 80 nanometers. This sensitivity enables EVs to be stained and analyzed directly in blood and plasma. However, small particle experimentation requires proper sample preparation and instrument operation. Debris in the sample or instrument will be detected as small particles, so it's important to keep them clean. If the sample is swarming, it may appear larger than normal or possibly detectable on an otherwise less sensitive detector or instrument. However, such data are combinations of multiple signals and multiple events and are artifacts. Titrating the samples and always acquiring multiple dilutions will help to ensure that optimal data are collected. The abort rate can also help to identify when swarming has taken place. And as mentioned, less than 10% is important and less than 5% is best. Different purification methods can increase the prevalence of specific EV populations. However, they can eliminate others. So it is important to cater your purification approach to the population of interest. Ultimately, thoughtful planning and careful attention to detail are the keys to effective small particle experimentation. Thank you all for attending. If you have any questions, please enter them into the Q&A box. Okay.
All right, so the first question for you is, how can I differentiate between particulate in the sample probe, or the sheath solution?
Okay, well, so this is actually quite easy. If there's particulate in the sample probe, depending on the rate that you flow it at, it's going to change the amount of particulate that's flowing. So you can just alter the flow rate, and you actually see more or less debris. If it's in the sheath, it would stay constant because the sheath rate does not change. So if after cleaning the probe you still have a lot of noise, it's most likely in the sheath. If both of these are clean and then you're analyzing your sample, then if you have a lot of debris in the sample, then that's where your debris is. Need cleaner buffers.
Okay, George, the second question is, what is the source of PBS that you use for small particles?
Well, so I've used a variety of sources. The one that I use most commonly is from Sigma Aldrich. Sigma is owned by EMD Millipore. They're the water purification experts, and they tend to have pretty clean buffers. But you can make your own even with 10x solution or even homemade PBS with, as long as you use clean water, it'll minimize particulate.
Okay, another one, does the broad distribution of EV populations affect resolution from background?
It can. So EVs aren't a mono, like really narrow dispersed population like a man-made particle may be. So as opposed to having like a really thin, narrow defined peak, they spread out a little broader. So if you're comparing signal versus noise of population versus, for instance, optical noise, it can it can make the population appear more like a shoulder sticking out from background if they're really, really small. So, but the resolution can be, or the identification of these particles can be definitely improved with immunophenotyping. In fact, it's necessary to immunophenotype to identify populations.
Okay, George, here's another one. Will VSSC improve detection on any flow cytometers?
Well, it depends. It can. But it depends on a variety of factors. So each laser on a flow cytometer has different, power output. They may have different widths across the core stream and different focusing. It changes the power output at that specific interrogation point. But in general, the power itself is a big factor. So, for instance, theoretically, a UV laser may have even better sensitivity, but most UV lasers are really weak. And in the case of most flow cytometers, by Snell's law, it actually would marginally shift the refractive index of particles and enhance detection. But for the CytoFLEX, there's a bigger enhancement because the side scatter itself is actually attenuated. So it makes a dramatic difference on the CytoFLEX, but may not make that much of a difference on any other flow cytometer.
All right, George, I'm just getting some other questions for you. Did you analyze EVs from bacteria in your system?
No, I have not. We've analyzed some bacteria, but not bacterial EVs.
Okay, can you label intra-EV markers?
Yes, you can, but to label intra-EV, you need to permeabilize the EVs. So some folks in literature use, simple like saponin-based detergents, and some people use like Triton X and such. But this can be a little complicated because it can increase the noise, so you might need to better purify your samples afterwards. But what may be even a little more complicated is there's not a whole lot of intra-EV markers that are available, or at least the interesting ones